Hi, everybody. Um, Questions from last time? Any comments, anything? All right. So I've shared the mid-semester exam. It's due in 3 weeks. And as you will find if you haven't already seen it, it requires you to read up on what are called Wright-Fisher debates on evolution, primarily exchanges between these two men, either via publications or letters to each other, regarding many aspects of evolution. But in this course, we want to focus on two. The first one is the nature of evolution. That's in the paper that I shared. It's called Controversy 2. And the second one is the debate on dominance. Is a debate on dominance. So to get a perspective of these two debates, you should get started with the paper that I sent you as the initial read, and then look up other sources which help you address the particular questions that I have asked in the quote unquote exam. And obviously it's open ended, and I'm going to judge submissions based on the most important being how much effort did you put in. And in something like this, it's extremely easy to sense when somebody is started on sixth evening when the debate when the submission was due on 8th. It's very easy to find out. So the most important criteria for me is how much effort did you put in, and that becomes very apparent once you read someone's submission. No right or wrong answers. If you just wonder about the questions and explain that this is why I can't comment on it, this is why I'm confused, these are the issues that are not clear to me, and you do it well, I have absolutely no problem giving you full credit for that. But what must come out is the effort that you've put in. So, I mean, needless to say, for something like this, if you start at the last minute, nothing's going to happen. All right. So today we will do something called a traveling wave model to capture adaptation. And I've tried to keep the math to as little as I can. But this work is from Daniel Fisher's group at Stanford, and this was published in Genetics in 2008. While the central arguments do not change in terms of what I will cover today, the paper has many more details, and it also presents analysis when some of the assumptions that we're going to make today. Excuse me. Some of the paper has analysis also for parts where some of the assumptions that we're going to make today are relaxed, and they study, how much do you deviate from the results that we get when we do it the way we're going to do today, compared to when you relax those assumptions. So it's a really interesting work, I think. But as I said, even if you don't, towards the end there is math, before that there is no math. I only want to develop intuition qualitatively and graphically. One of the things I think we should all learn to think about is how to think graphically. If we have a phenomena and we have two variables in that phenomena, x and y, then one of the things that we should let our intuition develop is that what sort of relationships do we expect between x and y, and just plot that graphically. The physics of it, the mathematical representation of it will come later. But what's very important is for us to understand what does our intuition tell us, and then find holes in it, that is this how it's going to play out, and so on and so forth. So we'll develop intuition intuitively. We'll get to some math. even if you don't get the math part is going to be at the end of the class, and even if you don't get all of the math, that's okay. You definitely need calculus for that. It's relative. If you know calculus, it's relatively simple. But even if you don't, no big deal about it. The idea is to get the spirit of the argument and not so much the nitty-gritties of the math. All right. So if there are no questions, so we're going to do this traveling wave model of evolution. From last time, we saw that clonal interference persists in a culture. And what we mean by that is that many beneficial mutations exist in a culture at the same time. And what that automatically means in an asexual population is that these beneficial mutations are going to compete against each other. And because you can't trade beneficial mutations with each other, only one of them is going to reach fixation. The others are going to get eliminated. So in that sense, most of the beneficial mutations are lost, not just because of drift, but because there are multiple beneficial mutations present at the same time, and many are lost. So imagine I'm doing an evolution experiment and I'm measuring growth rate of E. coli in a particular environment. And obviously, when I'm doing this experiment, mutations which confer a higher growth rate are going to get fixed. So the fitness will keep increasing with time. But if I were to do this for a really long time, what do you expect this graph will look like? The x-axis represents the time that I've run the experiment for, which can be captured in number of generations that I've processed. And y-axis is just the fitness at any given point in time. What do you expect this graph to look like? We touched on this last time, but I want to be more explicit today. Somebody.
It will be the same point as we discussed.
Okay.
For one single mutation, if there are more than one mutation, there will be multiple segments, and eventually only one of them will be dominant.
I am okay. Fine. So let me clarify. One is that this number of generations here is a time scale that's very long. Okay. So we are talking of a time scale which is much larger than the waiting time for a mutation is much larger than fixation time. So this is of a time scale that's really large. The second is that we are talking of, let's try and keep things simple. Let's say that we are talking of bacteria. So traveling wave model is actually for bacteria. So let's say that this is for bacteria. So there is no question of dominance or recessive alleles, right? There is only one allele. In that context, what do you guys think that the graph will look like? Somebody?
It'll increase and then become a constant.
Sure. Why does it become constant?
Because it'll reach the peak of the fitness landscape.
Sure. That's fair. Fine. So Yash, why do you say sigmoidal?
Um, yeah, it will take some time for the allele to reach the critical fixation, and after that it will grow logarithmically.
Well, I am not sure what you mean, because as we said, the time to reach fixation or the waiting time is much smaller as compared to this entire time scale, right? So those times don't matter here. And what we are comparing here is the mean fitness of the population, right?
Okay.
So what has been experimentally observed, and this has happened in many studies, is that almost without exception, this is what the graph is going to look like. So what Pavitra said, exactly that's what happens. So it starts to increase, but then it plateaus, and we'll discuss one particular paper post mid-semester exam week where this particular part of the graph led to some really interesting results. But for us, for today's classes purpose, what we are interested in is this linear regime. Okay? So the initial bit, at least this bit, this is more or less linear in the early part of evolution experiment. And what early means is dependent on which species you are working with, what were the environmental conditions in which this experiment was done. But this more or less linear regime holds true for at least a few hundred generations. So at least for that time scale, this is a fair assumption. And then we are going to say that the slope of this curve for this, so remember, I'm only talking about the linear part here, that for this part, the slope of this trajectory is the speed of adaptation, which, because the slope here represents per generation how much fitness gain happened. And now what we are going to, the question that we are going to ask is that at what population size n is the speed of adaptation maximum? That what should be the, suppose I'm interested in evolving a bacterial population the fastest I can. What we saw last time is that tau weight and tau fix are quantities which are functions of population size n. And n is something that I can easily adjust. And if I want my populations to adapt the fastest they can, then I would like to work with a n that facilitates maximum slope in this linear part. And hence, that's the question that I'm interested in, that what does the slope, how does the slope change as I change n in a bacterial species? Does that question make sense? Is that okay?
Yes, but I'm still not clear why it is linear.
Okay, yeah, sure. So why do you say sigmoidal? Say that again.
So we were talking about an allele which emerged in the population, and we
Which
want to
what?
see
Which what? I'm sorry, say that
emerged
again.
in the emerged
Emerged.
appeared. Yeah.
Sure, yeah.
So and we are talking about how it will reach fixation.
Right,
Correct.
right.
So initially it won't have that many partners to get prominent in the population, right?
No, but bacteria, right? No dominance.
Okay. There is no dominance.
There's no dominance. There is only one copy, right? And what's going to happen is that initially, early on, the way it plays out is the following: that early on, when you start the experiments, there are a lot of beneficial mutations which are possible. So, some member acquires one beneficial mutation, another member acquires another beneficial mutation, and so on and so forth. And all these beneficial mutations start competing against each other. But while they're competing and some of them are eventually going to lose, what also happens is the fact that the mean fitness of the population increases very rapidly because there are so many beneficial mutations in the population. So the mean fitness with generations increases very rapidly. As you move forward in the experiment, now beneficial mutations are drying up. There are not that many available. And graphically, you can think of it like Pavitra said, that you're reaching the fitness peak. Now, most mutations are actually deleterious. So, now because some beneficial mutations at least have reached fixation, and supply of new beneficial mutations isn't happening at the same rate, hence the rate of adaptation or the slope of that curve decreases with time, and that's why the manifestation looks like what we drew. Does that make sense? All right. Okay. So that's what we are interested in. The first problem that we encounter is the following: that not all beneficial mutations are going to have the same selection coefficient, right? That's obvious. Some beneficial mutations might confer an advantage of 1%, some might confer an advantage of 2%, and so on and so forth. So first of all, let me ask you this question. Suppose I have a population and suppose I have a population and suppose I have a genotype, and how many one nucleotide mutations are possible for this genotype? Let's imagine that the length of genotype is L. How many one nucleotide mutations are possible for this genotype? In other words, what I'm asking is that how many sequences are there which are at Hamming distance equal to 1 from this genotype? Namrata, you answered this question a couple of weeks back.
3L, sir.
3L. Does everybody say 3L? See 3L? Okay, fine. So 3L, right? So what that means is that I can introduce 3L. I can make 3L genomes which are at Hamming distance equal to 1 from the sequence that I have. Now of these 3L, let's imagine, and I'm making this up here, let's imagine that L are deleterious. So we forget about those. Of the remaining 2L, let's say another L are neutral, so I forget about those. But the last remaining L are beneficial mutations, right? And I'm making these fractions 1/3, 1/3, 1/3 up. Beneficial mutations could be 0.1 L for all we care, but that doesn't change what we're going to argue here. So we have L beneficial mutations. What I'm going to do in this hypothetical experiment is that for these L mutations, for these L beneficial mutations, I am going to measure the selection coefficient associated with each one of these L mutations. Okay, so some mutation will give me an advantage of 0.8%, some a big mutation will give me an advantage of 3%, and so on and so forth. So I have a list of L mutations, and I have a corresponding vector of what was the benefit conferred by this particular mutation. And then I draw a frequency distribution curve like this. And what I'm saying is, so this is S, so this is S, so this is basically 1%, 2%. What I'm asking is that what percentage of all beneficial mutations conferred an advantage between 0 and 1%? What percentage of beneficial mutations conferred an advantage between 1% and 2%, 2% and 3%, and so on and so forth, right? So I'm going to look, I'm basically what I want is draw this frequency distribution histogram for beneficial mutations. Is the question clear to everybody?
Yeah.
All right, great. So now tell me, what does your intuition say about the nature of this curve? What do you think this graph would look like?
Decreasing graph.
Who was that? Was that Rishi?
So
Yeah.
it will decrease
Why do you say that?
because the value of S will decrease because it won't be 1 for some mutations. For some it will be.
No, say that again. Value of S will decrease.
Yeah.
What does that mean? No, S is an independent variable here, right?
Yeah, but there won't be a mutation that will be 100 times beneficial than another mutation. So
Right.
some mutations are very less beneficial, some are more beneficial.
Right.
The number of less beneficial mutations are more than number of more beneficial.
Sure. Agreed. So what you are saying is that according to your intuition of how a cell would work, most beneficial mutations are going to confer a small advantage. Whereas finding a beneficial mutation which confers a large advantage is going to be relatively rare. So this graph is going to be decreasing in nature in some fashion. We don't know the exact distribution, but it's going to be decreasing. Sure. Anybody else? Okay. I think what Rishi has said is actually summed this up very well. So this distribution, whatever this graph might look like, you can think of this as histograms. For those of you who are more comfortable with continuous distribution of probabilities, you can think of this as a smooth curve also. This distribution, whatever it might look like, is called distribution of fitness effects, also called as DFE in the literature. And there's been a lot of experimental and theoretical work in this. In fact, one of the readings again after midsem is a paper from Roy Kishni. And remember the antibiotic video that we saw where bacteria were growing and becoming resistant and so on and so forth? That was from Roy Kishni's group. Another paper from Roy Kishni and Daniel Hartler, who is a population geneticist. There was a paper in 2007 in Science where they tried to measure this distribution experimentally, and they failed. And they failed because of a very interesting reason. And now we have all the theoretical understanding. Now we have all the analysis for us to study for us to understand and appreciate why they failed. Okay, and we'll do that in a couple of weeks' time. But this distribution is called DFE, right? And what the consensus is that DFE for beneficial mutations can be represented as some sort of an exponential distribution. So there is an exponential decay here, and it approaches zero pretty fast. So much more likely to have small beneficial mutations than mutations which confer a large advantage. Fine. Our solution, however, today is going to be that we are going to ignore DFE. We will say it doesn't matter. We know that there is a distribution. We're not going to worry about it. We will say all beneficial mutations have the same selection coefficient S0. Okay. Now this might seem extremely dissatisfactory. However, once we understand this part, extending it to incorporate for a DFE is actually not very hard then. Okay. So we will only do this, and I'll share the reference of the Fisher paper. So every beneficial mutation has selection coefficient S0. Additionally, we are going to ignore epistasis. Okay. Remember, epistasis is the fact that if one mutation confers an advantage of 1%, another mutation confers an advantage of 0.5%. When they are both present in the same genome, the advantage is no longer 1.5%, it's something else. Any deviation from additive benefits of beneficial mutations is epistasis. For the linear regime that we are talking about, for this linear regime, we are going to ignore epistasis and say that beneficial mutations have additive effects. Ignoring DFE and this assumption of a constant S0 is not a very seriously flawed assumption, because we are ignoring DFE partly because this is a first course in evolution and it's a more advanced course in population genetics and evolution, which would cover that. So that's not too problematic. Ignoring epistasis is problematic. There is definitely epistasis between beneficial mutations. However, how those interactions take place at a genome level when the entire cell is evolving, I don't know anything specific that's happening in a system, but I'm only looking at a bird's eye view of what is happening in the cell. that qualitative nature, or that even quantitative nature of epistasis, behavior of epistasis, is something that we don't know yet. So for this purpose, we ignore epistasis for now. So this is how it's going to play out in a population. Remember, we discussed two regimes last time. Regime 1 was where the tau weight was really long. If tau weight is really long, then at the beginning of my experiment, all individuals, and I'm going to assume that my population size is n, all n individuals are at fitness f0, which is the fitness of the ancestor. I wait a little while, this will be the tau weight, and then one individual picks up a mutation and escapes drift. And what happens as a result is that I have n-1 individuals here and one individual here. Right? Of course, by the time it has escaped drift, the number of individuals of this particular mutations are not is not 1, but it's actually 1 by S0. Because remember, we said that a mutation has escaped drift when there are 1 by S individuals corresponding to that particular genotype. So we only find out whether a beneficial mutation has escaped drift or not when these numbers reach 1 by S0 in this case. But if we go back, and some mutation would escape drift at some point, and if we look at when the first mutation happened, this is what the frequency distribution is going to look like. And now this mutation goes to escape drift and get established in the population and reach towards fixation. And as a result of this, frequency of the ancestral genotype is going to decrease, frequency of the mutant genotype is going to increase. And because there are no more new supplies of beneficial mutations in the population, this mutation reaches fixation. So we reach a scenario where there is no individual corresponding to the ancestral genotype and n individuals corresponding to this genotype which carries this one beneficial mutation. And then the picture stays like this for a while. Eventually, one of the individuals in this new genotype is going to pick up another beneficial mutation, and all beneficial mutations have a fitness effect S0, and hence this new genotype that's created will have a fitness F0 plus 2S0, and this story will sort of keep repeating itself. So in regime 1, evolution is going to take place. This is going to be the dynamics of how it's going to play out. In regime 2, it's going to happen slightly differently. Starting point remains the same. So this is F0, and I'm not going to write F0 from now on. So this is F0. We have n individuals at F0. First mutation appears, which escapes drift, and hence I have n-1 individuals here, one individual at S0. Then selection acts, and as a result of this, the ancestral genotype decreases in fitness, the new genotype increases in fitness. But now the difference that takes place from the previous setting is that before this guy reaches fixation and this is completely eliminated, another mutation takes place. And that another mutation to establish this 2S0 genotype has to take place in the middle one, because all beneficial mutations confer an advantage S0. It's also possible that one of these individuals picked up a beneficial mutation again, simply because there are so many of them. It's possible, especially in this regime where there are multiple beneficial mutations. It's also possible that one of these guys picks up a mutation, but it's going to land up here again. So while the genotype of the two beneficial mutations might be different, phenotypically they are identical. They've been lumped together here. So we are interested in the beneficial mutation that occurs in the fittest bar. And when the picture of the population is like this, the fittest bar is these guys which are at fitness S0. So one mutation happens, and one more mutation happens, and now we have an individual at 2S0. The frequency of ancestral will decrease, this guy will increase, this guy will increase. I have two questions for you. One, One, what does this look like when the profile fully develops? So it's obvious that this is a very dynamic picture and things are changing here every instant, right? So these are changing. Do we reach a stage where this profile develops? Remember, these guys are going extinct and newer mutations are coming into picture. So what does this picture look like when the profile is fully developed in that linear part of the graph? And in that fully developed profile, which genotypes are increasing in frequency and which genotypes are decreasing in frequency is the second question. Okay. Is the question clear to you? Yes, no, something?
Yes, yes.
Anybody has a question? Because I want to give you guys 2-3 minutes to just think about it. What does the fully developed profile? Because if you understand that if you understand that intuition, if you understand that intuitively, that makes the analysis thereafter a lot more easier. Any questions with this?
What do you mean by fully developed here, profile?
Yeah, it's not very clear. Fully developed means that does this profile reach a steady state? Is there some feature of the so clearly this is changing, and our starting point was something which was sort of very abnormal in the sense that a single genotype in a regime where multiple beneficial mutations are going to coexist. This is not going to be a steady state picture. By fully developed, I mean, can you imagine a scenario where the starting point given this, let me rephrase that. Can you imagine a scenario in this setting where eventually you reach a state where newer genotypes are arriving at a certain rate because of new mutations, and the older genotypes, the least fit genotypes, are being eliminated at the same rate? Can you imagine that scenario? Yes or no?
Yes, so
Anirudh?
but it seems like more of ideal.
Doesn't matter ideal. We will do ideal.
Okay, sir.
Anirudh, does that make sense?
Yeah, yeah.
Right? So you have newer genotypes arriving because of mutations. You have old genotypes being eliminated because of selection. When I say fully developed profile, what I mean is that these two rates match each other. Okay.
Yeah, yeah.
Yeah. So everybody just take maybe 2-3 minutes. I will just take a short break here and just tell me what you think the fully developed profile looks like. And fully developed profile happens at a time when genotypes are arriving because of mutations and leaving because of selection at the same rate, right? So I'll just give you 2-3 minutes and then we'll pick up from there. All right, what do you think?
I think it would be a normal distribution.
Why do you say that?
Because at a time there are more than one mutations competing. The old ones are depleting in number, new mutations are increasing in number, but there will be one mutation which will be taking the mean place. I mean, the mean fitness will be represented by one or two
Right,
individual mutations.
right, right. Anirudh?
Yeah, pretty much the same thing, but I think that it would be moving
Mm-hmm.
toward the right in the sense that the
Right.
mean keep changing.
Absolutely. Yep. Anybody else? Yeah, yes, go ahead.
Yeah, just that I agree with that too.
Yeah. Sure. Others? Gaurav, what do you think? Gaurav is not awake. All right, perfect. I mean, between the two of you, that's exactly what happens. That this is what the picture is going to look like. We have to imagine it this way, that new mutation happens, another mutation happens, and now at this point in time, this genotype is definitely increasing in frequency. Leftmost genotype is definitely decreasing in frequency. What is happening to the frequency of the middle genotype? As in the snapshot that's shown at this particular instant, what do you think? Is the frequency going up or down of the middle bar?
But the rate of decrease would be increasing. The rate of increase would be decreasing, I guess.
But why do you say increasing?
I don't know.
Other than the fact that there is an arrow there, which is going up. I guess what I'm trying to get an answer out of you guys is that suppose we have this distribution of genotypes. How do we know which genotype is increasing and which is decreasing? There are only 3 here. So it's relatively easy. The fittest one is obviously increasing. The least fit is definitely decreasing. How do we say about the middle graph? Anybody? So, I mean, this can be worked out quantitatively pretty easily, but it can be shown that any fitness level which is above the mean fitness of the population is increasing in frequency, and any fitness level which is below the mean fitness of the population is decreasing in frequency. So as the snapshot of the picture is shown in this figure, the mean fitness, so this is F0, this is F0 plus S0, this is F0 plus 2S0. It's fairly obvious to see that the mean fitness of the population at this instant, so this is at S0, there are very few individuals with 2S0 fitness. We can just call it 0S0 and 2S0. The F0 is common for, so we can remove that. The mean fitness of this population is between 0 and S0, right? Does everybody see that?
Yes, sir.
Yes, sir.
Cool. So the mean fitness of the population is between 0 and S0, and hence S0 at this particular instant is fitter than the mean fitness of the population, and hence its frequency is going to increase. And as Anirudh said, the frequency is going to the frequency is going to increase at a rate which is proportional to how far you are from the mean fitness. So the greater the advantage over the mean fitness, the faster you are increasing in frequency. So if we play this out a little further, what's going to happen is that this is going to decrease, this is going to increase, and this is going to increase. And now, as that is happening, we will reach an instant where the frequency of this and frequency of 0 and frequency of 2s0 is equal. At that particular instant, the fitness of the mean fitness of the population is also s0, and hence fitness of this genotype is equal to mean fitness. And at that instant, its frequency will no longer change because it's outcompeting this guys at the same rate as it is being outcompeted by this particular genotype. So frequency no longer changes. And if we play that out a little more forward in time, frequency of 0 decreases, frequency of 2s0 increases, and now mean fitness is greater than s0, and hence then this will start decreasing in frequency also. Okay? So your relative frequency increases or decreases depending on what is your fitness level relative to the mean fitness of the population. All right. So between the two of you, you have everything covered. That at some sort of an equilibrium, not equilibrium that this picture is static, but equilibrium in the sense that this shape remains constant, this is what the population is going to look like. At the leading edge, this part of the curve of the distribution is called the leading edge. At the leading edge, individuals are few. That's because these individuals represent mutations which came up only recently in the population, and they just haven't spent enough time in the culture such that they increase in frequency. But of course, their fitness is well over the mean fitness, and hence all these guys are increasing in frequency. At the other end of the spectrum are genotypes which are being eliminated from the population. And when the shape reaches a steady state, what is going to happen is that this distribution is moving to the right at a constant speed. So this is called the leading edge. The fittest genotype is called the leading edge of this wave. So this is now this distribution is moving to the right, and hence it is called a traveling wave. Its shape is not changing, but it's simply moving to the right. And the movement of right, when the shape has reached a steady state, looks like this. That this is called the leading edge. The fittest genotype in the population represents what is called the leading edge. It is up to these guys to pick up a mutation and establish another leading edge of the population. When that happens, then this is no longer the leading edge because we have a fitter genotype here. That, of course, is happening at a certain rate. And the rate of this happening, so when that happens, the wave moves 1 unit to the right. That rate is exactly matched by the least fit genotype being eliminated by selection. So this distribution goes to 0. This particular genotype reaches the level of this particular genotype. So this shrinks to this one because of the action of selection. This goes to this much because of the action of selection, and so on and so forth. And it's almost as if this wave moved 1 unit to the right. Hence, it is called a traveling wave model. Its shape does not change with time. Does this make sense?
So just
Yes.
does the mean move? Is the movement of mean steady in this case towards the right? Is it always going to be the same? The rate of movement of the wheel?
Yeah. In this context, yes, right, because we are saying that this matches with this, right? And the mean is going to constantly move. So that's actually the next part that I'm saying. So this traveling wave of adaptation model, what we are interested in is the speed at which the wave is moving to the right. and that speed is the rate of adaptation, right? So how much when the wave moves, let me ask you this question. When the wave moves 1 unit to the right, by how much does the fitness increase? By how much does the mean fitness of the wave increase?
1s.
S naught, right? Because every individual has picked up S naught units in fitness, right? Because the wave, not technically, it's not as if every individual has moved to the right, but the frequencies have so changed that you can imagine that every individual has just moved S naught units to the right. So the mean fitness has, with every rightward movement of the wave, mean fitness increases by S naught. And what we are going to be interested in is that what is the time tau in which this wave moves 1 unit to the right. Okay. If we get an estimate of what is the time tau taken for this wave, taken by this wave to move 1 unit to the right, then S by tau, because fitness increases S, time taken for that fitness increases tau, S by tau is the rate of adaptation that we are interested in. Right? And that's essentially what I have done here. That if the wave moves 1 unit to the right in tau time, then speed of adaptation is simply S naught by tau, where S naught is the increase in mean fitness of the population and tau is the time for that increase. Another problem with this traveling wave model is that we don't know the width of this wave. We don't know how many mutations exist in that population. Right? If we assume that the front or the leading edge of the wave is k beneficial mutations ahead of the mean. So if this is the mean fitness F naught at this particular instant, then the leading edge is F naught plus k times S naught. So this is k beneficial mutations ahead of the mean, right? And if I know k, I know some sort of I have some idea of the distribution associated with this traveling wave. Our job next is to find both k and tau. If we know k and tau, then we know a lot about what is happening in the population. And next, I think I'm done with all the points. Yes. We're going to start with the math part now, but is the intuition behind this clear now? People who haven't spoken up. Rachita, Pavitra, Shriya.
Yes, sir.
Yes.
Sir, so in this model, we aren't waiting for any one mutation to get fixed, right?
Yes, we're not. We're not. Yeah.
Okay.
Yes.
Yeah, then clear, sir.
So,
Yeah.
there are two terms, right? Speed of adaptation, rate of adaptation. Are they different?
No, they're the same.
Same,
They're the
right?
Yeah, same. All right, cool. So the math is not terrible, and I don't think understanding the math is also hard, but I think coming up with this analysis is really at least why I like this paper so much. The logic is as follows. If the wave is moving to the right every tau time, then two things will happen, right? And you have to make sure that you are comfortable with both. So remember, in tau time, the leading, so we are going to assume that it's smooth motion, it's moving like clockwork. Of course, it doesn't happen like that. We're talking of statistical averages. The larger the population, the greater the regularity associated with the evolutionary response of a population to an environment also. All right. So population is moving like clockwork to the right on this graph, right? And it moves 1 unit to the right every tau time. If that is the case, then two things will happen. in tau time, the leading edge will no longer be the leading edge. If this is the leading edge right now, so I've made this picture with k equal to 3, but we have no idea about the k. Right? K is something that we are interested in, we want to find out. Suppose this was a case where k was equal to 3, and if this is what the picture looked like right now, after tau time, I tried making these dotted lines, but they aren't clear at all. But basically what I'm saying is that in tau time, this leading edge will no longer be the leading edge, and this leading edge has grown up to become this second bar here, and we have establishment of a new leading edge, which is this. So this happens in tau time. The other thing that we note is if this was the leading edge now, after k tau time, and remember k is the number of beneficial mutations that the leading edge is carrying compared to the mean fitness of the population. After k tau time, this leading edge will become the most frequent genotype in the population. Right? Are these two things clear?
Yes, sir.
Yes, sir.
All right. Cool. And now we're going to play with this, that we're going to analyze these one at a time. Right? We'll consider this first. The number of cells in the leading edge, because it has become established, is 1 upon ks naught. Because remember, the selective advantage that the leading edge enjoys over the mean population is k times s naught. Over the mean fitness of the population is k times s naught. So that's the number of cells in the leading edge at this particular point. The growth advantage over population mean is k times s naught, right? Because population mean fitness is 0 in a scaled analysis of fitness. We'll call this 0 these as negative fitness, because everything is being scaled by the mean fitness. So growth advantage is k times s naught. Hence, the number of cells in the leading edge are growing at basically n into e to the power rt, where r is the relative growth advantage. This is just exponential growth model, and I mean this is just standard exponential growth of bacterial populations. This is the starting number of individuals, and this is e to the power rt, where r is the relative growth rate here. So the number of cells in the leading edge are growing like this. Now, leading edge will no longer be leading edge when one of these individuals picks up a mutation and the resulting beneficial mutation escapes drift, right? When these cells are growing, this is the number of cells, right, at any given point in time. This is the mutation rate, mu b, of beneficial mutations only. What does this expression give you?
Can you tell me what mu b is again?
Mu b is the rate of beneficial mutations. So this is number of individuals, right, at time t. This is mutation rate, the rate at which beneficial mutations occur, right? So this is sort of the total number of beneficial mutations occurring. You multiply that with dt, which basically tells you that in dt time, this many number of beneficial mutations are going to occur. I'm still not talking about this ks naught. Does that make sense to everybody?
So can you repeat that again?
Yeah, so this is the number of individuals at time t, right? In dt time, this many mu dt mutations are going to occur. Does that make sense?
Yeah.
Right. So for this many individuals in dt time, mu times dt, because mu is the mutation rate per cell per generation. So this many cells and this many generations, t can be measured in generations, and hence you get the total number of mutations, total number of beneficial mutations occurring in time dt. However, we know that not all beneficial mutations are going to survive. Most beneficial mutations are going to get lost because of drift. When a new beneficial mutation happens at this leading edge, what is the chance that that new beneficial mutation has survived drift? Come on, guys. When the leading edge picks up a new when one of the members of the leading edge picks up a beneficial mutation, right, what is the chance that this beneficial mutation survives drift?
So the frequency has to reach k plus 1 S naught. The number of population.
1 upon k plus 1 S naught.
Yeah, 1 upon k plus 1.
Yes. But what is the chance that, what is the probability? So remember we said that a mutation which has fitness advantage of S has a probability S of escaping drift, correct?
Yes, sir. So here k plus 1 is not.
Exactly. Yes. Right. So when this mutation happens in the traveling wave, when this mutation happens and we have a new mutant here, this one mutant, the relative advantage over the mean fitness of the population, we'll assume that because there is only one new mutant at this k plus 1 S fitness, k plus 1 S naught fitness, because there is only one, the mean fitness has not changed. The mean fitness is still there. So the relative advantage over the mean is k plus 1 S naught, right? So in time dt, the number of beneficial mutations that have occurred is simply the number of individuals at that particular times mu dt. But of these mutations that are happening, not all will survive drift, and technically it should be k plus 1 times S naught, because this new mutation enjoys a benefit of k plus 1 times S naught. But just math becomes a little more tractable if you just write k S naught, if we ignore the 1, right? So if k is 7 in a typical bacterial culture, putting k equal to 8 is not going to make a huge difference, right? But you're absolutely right that this should be k plus 1. Does this make sense? So if I integrate this over 0 to tau, then I get the total number of beneficial mutations which have escaped drift at the leading edge. Does that make sense?
Yeah.
Questions? I cannot believe that this has made sense to everybody, so you have to ask questions. Okay. All right. So this integral here is representing that in tau time, how many beneficial mutations survived drift at the leading edge. But from our from our modeling of traveling wave adaptation, in tau time, how many beneficial mutations escape drift at the leading edge by definition?
1.
1, exactly, because we are saying the speed of movement of the traveling wave is such that at every tau time, the wave moves 1 unit to the right. Absolutely, perfect. So this expression has to be equal to 1, right? So this expression is equal to total number of beneficial mutations at the leading edge which escape drift. This is just the number of individuals at the leading edge at time t. Mu dt gives me the number of mutations happening at the leading edge in time dt. Ks naught, multiplying it with Ks naught gives me what fraction of these mutations occurring in time dt escape drift, and then I integrate it over 0 to tau, which gives me total number of beneficial mutations at leading edge which escape drift over time tau. But we know that in time tau, leading edge is no longer the leading edge, hence the expression, this expression is simply equal to 1. So if I plug this equal to 1, then I get, it's just calculus. I get something like this, and then I just ignore this -1, assuming that this is large enough. We'll come back to that depending on what does k and tau turn out to be. We still have no idea about what are k and tau. And eventually, I mean, this is just algebra. You guys can just work it out yourself. We get tau is equal to something there. Let's call this equation 1. So we got an expression for tau, but remember that this expression for tau contains k, and k is still an unknown. So I really haven't figured out tau now. But now, to get the second equation, this is equation 1. To get the second equation, we will go back to this analysis, right? So far we focused on this part, that after time tau, leading edge is no longer leading edge. Now I will go to this analysis, that after k tau time, the leading edge actually is the most frequent genotype in the population. And this analysis is actually, at least I think, fairly straightforward. Okay, second part. For the first tau times, so remember what we are solving is that in k tau time, leading edge is the most frequent genotype. To analyze this, for the first tau time, the number of cells in the leading edge are growing as 1 upon ks naught. This is the starting number of cells in the leading edge, into e to the power ks naught, which is the relative advantage, times tau. Does everybody see that? Is everybody okay with that?
Yes, sir.
After this tau time, the leading edge is no longer the leading edge, right? As we just saw. And the relative advantage of this particular genotype is no longer k times s naught. It actually has reduced to k minus 1 times s naught, because it's s naught units closer to the mean fitness of the population, because the wave has moved. So for the next tau time, its numbers grow as the following, that in the first phase, when the advantage over the mean fitness was ks naught, at the end of that phase, its numbers were this much in the bracket. And for the next tau time, the advantage that it enjoys over the mean fitness is k minus 1 times s naught, right? Does that make sense to everybody? If that's clear, we're done then. Any questions with that? Clear to everybody? Not clear to everybody? Guys, you have to say something.
Yeah, I think it's clear.
Okay. So the idea is pretty simple. That, where did it go? The idea is pretty simple, that at this, when you have this leading edge, for the first tau time, the advantage that these individuals enjoy over the mean is actually k times s naught. So for the first tau time, its numbers are going to grow as an exponent of k times s naught tau, right? Because for the first tau time, its advantage is k times s naught. However, after time tau, a new leading edge has appeared, and these guys, which used to enjoy an advantage of k times s naught, only enjoy an advantage of k minus 1 times s naught for the next tau time, right? So for the next tau time, its numbers are going to grow as this expression, that this was the number of individuals in that genotype after time tau, and for the next tau time they are going to grow as k minus 1 times s naught, and for the next tau time they are only going to grow as k minus 2 times s naught into tau, and so on and so forth. At the time when the leading edge becomes the most frequent genotype, the number of cells of that genotype are given by 1 by ks naught into e to the power ks naught tau into e to the power k minus 1 times s naught tau, and so on and so forth, going all the way to e naught s naught tau. E naught s naught tau will be the last interval where this was the leading edge. What used to be the leading edge is now this particular genotype, and the relative advantage that this genotype enjoys over the mean fitness is simply s naught. So this is going to grow as e to the power s naught for the next tau interval, and hence the numbers are going to grow as e to the power s naught tau. Okay. So after k tau intervals, the number of individuals in the population, the number of individuals in what used to be the leading edge will be nt. I sum all of these up, so the exponents get added, and I get 1 upon k s naught into e to the power s naught tau, and I start counting from back 1 plus 2 plus 3 plus k minus 2 plus k minus 1 going all the way to k, and this is just a simple sum, which is e to the power s naught tau, k into k plus 1 by 2. Does that make sense to everybody?
Yeah.
What we have done here, what's important to realize is taken two different things, and we have taken two different solutions and stitched them together. At the leading edge, for the first analysis, when we said that in time tau, the leading edge will no longer be the leading edge, that is an event which is totally based on probability. It's a very stochastic event that could happen anytime in time tau. So we have taken sort of a statistical average of that stochastic process, right? And for the second approach, when we said that the leading edge now will be the most frequent genotype in k times tau time, that there is no stochasticity in that because numbers are large, because that the leading edge is already established, hence there is no randomness in that behavior. So that we modeled using deterministic exponential growth. And we know that one process takes tau time, leading edge to not be the leading edge anymore. That's a stochastic event. And the other process takes k times tau time, which is a completely deterministic event. So I will have one expression for tau time and one expression for k tau time. And I'm just going to compare the two and solve for k and tau. Before we do that, we don't have anybody from physics here, but I mean, this paper came out from people in Department of Physics, and I think physicists think a certain way that neither engineers nor biologists think, and they know how to make a simple assumption. And whereas people typically from engineering and sciences background, we will look for that complicated solution and not make a simple assumptions just to be ordered to be more accurate. But in biology, you don't have to be. Accuracy has different connotations in biology than compared to physical sciences. If you get to an answer within 15-20%, especially for an analysis which is as abstract as this, that's actually a wonderful, wonderful job. You don't have to complicate things in order to reach for that final bit of accuracy. And there is elegance in that. So in the paper, they do two certain things. Right? They do two things that I want to discuss. The first is pretty straightforward. I hope all of you got how we reached this. Right? Even if you didn't get what happened here, imagine that we reached this particular stage. Now we have to proceed further. And I think as most of us are engineers, some are biologists. What we would do is we will take NT this and then sum up all the exponents and get this particular expression. Right? Now, when I open this up, so presumably I'm going to take a log at some point because I want to get rid of this exponent. When I do that, I will have a very ugly looking log of k square plus k. Right? That's going to be difficult to deal with. What they do in the paper is actually really, really nice. How many intervals are there? Answer fast. How many intervals of time tau are there?
Okay.
K intervals. What is the advantage that I enjoy in the last interval? Somebody?
S naught. Right?
S naught. So there are k intervals. The advantage that I enjoy in the last interval is S naught. What is the advantage I enjoy in the second last interval? 2S naught, right? The advantage that I enjoy in the third last interval is 3S naught, and so on and so forth. And what is the advantage that I enjoy in the first interval? That's just k times S naught, right? So I start with k times S naught and end at 1 times S naught. What is the mean of that? K by 2 times k by 2, right? So what they do in the paper is simply make this assumption that instead of doing all this, I can simply think of this as if I have k intervals, and in each one of those intervals, I have an advantage of k by 2. Right? That leads me to this much more simpler exponent, which is going to be much easier to deal with later on. Right? Even if you didn't get this, don't worry about it. This is perfectly fine. The other issue I have is, while I have this expression that in k tau time, right? So you can use this expression or this expression. Don't worry too much about it. I don't think I did a good job explaining either. So what I have done is found out an expression that after k tau time, the number of cells in what used to be the leading edge is now the most frequent genotype of the population, and number of cells in that genotype is going to be this. But what is the other side of this equation? What should I equate this to? And I want you to comment on this. Is the problem clear? What I have found through this analysis is what is the number of cells in k tau time of what used to be the leading edge and is now the most frequent genotype in the population. What I have found is an expression of the number of cells in that bar. But what should I equate to? I don't have an expression. I don't have an equation. I simply have an expression for something. I need something here to make an equation out of this. What should I do here?
Isn't that the n of mean?
It is the n of mean. It is the number of individuals which are at mean fitness. But what is that? Is the problem clear? That I don't have the corresponding other side.
You can estimate it, right?
How?
Uh,
That's what we just did.
yeah, okay.
But what do I equate this expression to? Remember, I need to solve k and tau. That's my goal, right? Because S naught by tau is going to give me the rate of adaptation, the speed at which the wave is moving, and K is going to give me the constitution of the population in terms of the genetic diversity that exists. So I need two equations in K and tau. I got the first equation from the other analysis, which said that in time tau, leading edge will no longer be the leading edge. But I need another equation from this analysis. So I found an expression for number of individuals in the most frequent genotype. But then what is it equal to? What would you plug in?
The carrying capacity of the population?
Carrying capacity is n, right?
Yeah.
So if we go back to our notes, remember that this happened. Remember that when we made the wave, this entire sum is n, right?
Yeah.
Right. So what do you think? Is that a good substitution? Pavitra, let's continue that.
No, it's not.
Why is that not a good substitution?
Because the sum of all the types of organisms will be the carrying capacity.
Correct.
Yeah. So it won't be the right thing to do.
Okay. Others? Any ideas? Anything? We're stuck here.
If we continue for k tau time once again,
वो तो जीरो आ जाएगा. नहीं?
yeah,
So let me, okay, any other points? Let me tell you what they did. So you should be pleasantly surprised, Pavitra, to hear that they did this. What did they do?
They assumed that the other organisms were very small in number.
Why is this an okay thing to do?
is it because their number is decreasing?
Well, no. What do you mean? It's not decreasing yet, right? I mean, even if it's decreasing, you are concerned about what is the number currently, right?
Yeah,
And that's the most frequent.
right.
I don't think it was the best thing to too.
Okay. Okay. Why do you say that?
I don't know.
Hmm.
So I think first we'll have to just see.
Hmm.
Uh, I don't know. It just doesn't seem right.
Yeah, no, I agree. I agree in the sense that I think the dissatisfaction with it is totally justified. However, the argument is as follows: that let's imagine that the total population is 10 per 8 or 10 per 9, doesn't matter, 10 per 9, right? These guys, this bar is of the order of 10 per 10. I'm sorry, 10 per 9. This bar is of the order of 10 per 8. This is 10 per 7. This bar 10 per 6. This is 10 per 5, right? So as far as relative frequency is concerned, this is log scale, right? So this gives you a very unrealistic. So there is a difference of like 10 times between this bar and this bar. That's the data that we have from real growth experiments of bacteria. So this genotype is 10 times more frequent than other genotypes. So what you are saying actually is that it's this falsely drawn, or this picture drawn on a linear scale gives you an impression that this bar is maybe, I mean, I don't know, maybe 25% of the total population, whereas that's not the case at all. This is 10%, this is 1%, these are negligible. This is 11%, that's 11%. So this is actually at least 3/4 of the entire population. Anirudh, what do you say to that?
Yeah, like you said, I think it's in the same order of the total number of n, right?
Yeah, exactly. And again, physicists, they think simple, okay? We complicate things. And it's good enough. If it's 75%, I personally don't have any problem with this expression, this substitution then. Then you are saying that, ballpark, this could be 0.75 times, and it doesn't really make any difference. As long as you get the order of magnitude right, that's fine. So this gives you the expression of tau, right? So you have one expression, one relationship between tau and k, assuming done with a deterministic analysis that in k tau time the leading edge will be the most frequent edge, the most frequent genotype, and you have the other expression which we called equation 1 somewhere, equation 1, another relationship between k and tau, that in tau time the leading edge will no longer be the leading edge, right? So, and then you can solve these, and for various values of n, we can calculate tau. So we are interested in the value of n where tau is minimum, right? Run out of time, but I wish we had like half an hour or something. Not possible today. Tell me your. Obviously, there are many issues with this, but the point is not so much to capture every nitty-gritty and every complexity associated with the biological process. The point of this work is trying to establish a framework in which we can study a process as complicated as that. Right? In fact, just last year from the same group, they published a paper which directly provides for the first time evidence which is contrary to traveling wave model of adaptation. And what they show is that because we are assuming here that leading edge is, so evolution here in this model, evolution is contingent on one of these guys picking up a beneficial mutation. These guys picking up a beneficial mutation is irrelevant as far as evolutionary change is concerned, because you're not moving ahead. But because these guys are so few in number, the chances that one of them is going to pick up a beneficial mutation, which are not going to be very frequent in supply, is actually very small, right? So this picture provides, this analysis provides a picture of evolution which is actually slower than how it would play out in real terms, because we discussed distribution of fitness effects, because the actual distribution is not as if every mutation provides a 2% advantage. The actual distribution is something like this. There are mutations which might provide a 5% advantage also. Granted, not very many, but there are these mutations. So just last year, there was a paper in Nature by the same group which provides, where did that wave go? Which provides evidence that you have a traveling wave in a population, right? Maybe not as accurate as this, but that's not the point. The point is getting this conceptual framework in our head. Then we can tweak it according to realities. So you have a traveling wave going on. And then suddenly what happens is that this guy here, one of these individuals, picks up a huge beneficial mutation and jumps right up front, right? So you have this complexity now in a traveling wave of evolution not just being restricted to the leading edge, but now everybody can pick up a beneficial mutation and jump up and become a leading edge. And now we can think of analysis where we can tweak this model, where S is no longer constant now. And now everybody could participate in bringing about evolutionary change and jump right up front and establish itself to be the new leading edge, and so on and so forth. And that, of course, is going to speed up adaptive response of our population. Okay, we run out of time. Let's stop today, unless there are minor comments. I will see you guys in 10 days, whatever, 10 days time. And yeah, should you have questions about the midsem, our midsem, just feel free to write to me. And if I think it's a generic enough question, I will reply to everybody. Otherwise, I'm happy to respond to you. All right, if feel free to leave, or if you have minor questions, we can discuss them now.
So could you extend the submission deadline for midsem?
Extend the, I 3 हफ्ते और ज़्यादा चाहिए.
We already have a midsem between, and we need 1 week to prepare for the midsem, so we already lose 2 weeks maybe.
I'm sorry, midsem तो next week है ना सिर्फ?
So actually, our one midsem is getting postponed. One of the exam is getting postponed, so it's maybe between 4th or 8th.
How many midsems do you have?
I guess.
Huh?
5 or 6.
नहीं, वो postpone हो रहा है तो that's also going to free up your time in the next one, ना? And why should I accommodate somebody else? I don't really understand. If वो उसने schedule नहीं किया before 2/8, you ask that person to schedule it after 8. Now you say we have another one at 8. I don't find that a very convincing request. I usually am very liberal with time. I don't really care about these things, but this is too much. 3 हफ्ता दिया है. Anything else? All right. See you guys in 10 days time then.
Sir,
Hmm.
yeah, sir, I'll just mail to you to get the paper from it.
Oh, achha, sure. I will send it to you. Yes.
Yes, thank you, sir.
Bye.